Share my screen. Everybody see my screen? Yes. Good. Okay. Let's see. Okay. So last week we talked about Mendel and his experiments, right? Uh, just a quick review. His experiments, what he found was that parents had two copies of a gene, which again he didn't know it was a gene at the time, but he had two copies of a particle, and that particle was being passed on to children or offsprings. And when they were passed on to offsprings, only one of the genes from each parent was passed on. That way, when you get to the child, they both arrive with the same number of two. So to understand genetics or genes, we need to understand where they're coming from. Okay. So your genes are actually going to be held inside of DNA, and DNA is going to be found inside of cells. And with us, especially in other eukaryotes, they're found inside of a nucleus. So this is what you should have today. So this is a typical eukaryote. And when I say eukaryote, I mean that when I see the cell, I look right here. Up here now. So this circular structure right here, this is showing me a nucleus. And that nucleus inside of it is DNA. Not every organism or every cell on the planet has a nucleus. Ones that don't, they're called a special type of organism called a prokaryote. They still have DNA, but their DNA is not held inside of a nucleus. So whenever you activate genes, the all this material you see inside this nucleus, this is all DNA. And then every individual is different. Every organism is. They have different number of chromosomes, different number of or amount of DNA, and not everything is going to be activated in, or not all DNA is going to be activated in every cell. So if you look up here in the corner, these are all different types of cells you would find in the human body. So this guy right here, the square one, this is called a simple cuboidal. The one in the back, that long one, so it's going behind the box, that's called a neuron. So that's a nerve cell. This one right here is actually a multinucleated cell. This would be a skeletal muscle cell. So this is what you use to move your muscles with. These two right here are red blood cells. When they're formed, you actually don't see a nucleus. So these dots you see into the cells, those are nuclei. But over here with the red blood cells, they actually lack nuclei because when they're formed, we get rid of the nucleus. That way we can pack more oxygen in it. And that guy in the background, that spiky one, that's an immune cell. That's a white blood cell. Or yeah, it's probably a white blood cell. But it's gonna be fighting off diseases or bacteria in the body if you were to get sick. So each one of these cells, if it came from the same person, would have the exact same amount of DNA, exact same chromosomes. But depending on what the cell wants to do, it'll pick and choose what genes to activate. So this square cell picked cells that made it a square shape. This one here picked cells that made it elongate and form specific proteins. And this one here picked genes that made it flat and disc-like. Um, the way we regulate genes is very complicated, but you just basically have factors inside of cells that can turn on DNA, and if those factors are activated, specific DNA segments are going to be transcribed and those proteins are made. Let's see. So some other structures that are important for genetics. We have this structure here. So you see how the nucleus is kind of connected to this structure. So this is called a rough endoplasmic reticulum. And the reason why we call it rough is it's got these little dots on it. And the word we use is studded. And studded means it's bumpy. And these ribosomes are going to be very important for making proteins. So if you look here, these little lines that are coming off the ribosomes, these are proteins being made by the DNA that's inside of the nucleus. So in each cell will pick and choose what specific DNA it would want to make. We also have ribosomes free floating. I don't know if there's any in this image, but every now and again you see little dots. So these little dots that are also inside the cytoplasm, they can also make proteins. The mitochondria is important. You might have heard of this one, but the mitochondria is going to be the powerhouse of the cell. It's what makes energy. So it makes a specific molecule called ATP, And ATP is like our currency for energy. If you want to do work, you spend ATP in order to get that work done. Let's see. It shows you other structures that you might not need to know for genetics, but centrioles right here, these little flower-looking structures, they move organelles. So these are really important for mitosis later if we get to mitosis. This look down here is the Golgi apparatus. It's going to be the kind of like a post office. It receives proteins, and then when it receives the proteins, it sorts them into different areas, and then it'll ship them to specific parts of the cell. The plasma membrane is important. So it kind of looks like this. Uh, let's see. It's a good word for it. Uh, I guess lineated structure, but you can see it has two barriers. One barrier is over here, the other barrier is over here. So this is how we keep things on the outside of the cell, on the outside, and then we things that are on the inside of the cell on this internal slide on the inside. So we're not able to freely exchange the external environment with our internal environment, and that's going to be very important for keeping organelles in place, keeping water concentration normal. Let's see. So this is the list I sent out earlier this week. So going over these names. The first one here is a prokaryote. So whenever you see karyote, that means a cell, and then a prokaryote, in this case, means a cell with no nucleus. So down here, this one first. This guy right here has a big circular nucleus. So this is not a prokaryote. Going up to this guy doesn't have a nucleus. It has DNA, so again, all that stringy looking structure, that is DNA, but it's able to free float around inside of the cell. So this guy right here is actually going to be a prokaryote. And then prokaryotes, there's two types. You've probably heard of bacteria before. So bacteria are prokaryotes. And there's one other classification of prokaryotes. They're called archaea. Archaea are very similar to bacteria, but you find them in areas where you wouldn't expect individuals. Like if you were to go to hot volcano beds, there would be bacteria there. If you were to go to poisonous gas swamps, there would be bacteria there. So those types of bacteria called archaea live in those extreme environments, and we call them extremophiles. The cells do have similarities. So you can see that it does have a cell membrane. And if you look down here on ours, we actually have a cell membrane. Bacteria should have a cell wall. So the cell wall's job is to keep the shape of the bacteria. So you see how this one's elongated? We call that a rod shape. And then that cell wall helps it maintain the that oblong rod shape. And then finally, it does have a glycocalyx or a sugar coat, this last layer here. This is actually protecting the bacteria. So if you were to have a bacterial infection, the reason why it's so hard to get rid of sometimes is this last layer here. It's protecting the bacteria. So if you wanted to actually get rid of the bacteria, if you were to weaken this layer and thin it out, it's going to actually make it more easier to kill inside of your body. When you're looking at sizes, these are not to scale. This is a eukaryote. I'll get to this in a second. But with the prokaryotes, they're roughly a tenth the size of a eukaryote. So if these two are actually together side by side, the prokaryote would be about that size, while all eukaryotes are roughly 100 times bigger. Okay, and then we have eukaryote. So a eukaryote, if you don't know the prefix, it usually means true. So a true cell. And then right here, again, that big structure, that's a nucleus. So that nucleus is going to make this cell a eukaryote. Now with eukaryotes, there's a larger array of organisms. So all plants, animals, and fungi are going to be eukaryotes, meaning that they have a nucleus if you were looking at their cell. There's also another type called a protist, but we won't even worry about protists. And again, kind of was on this is what's in your book, right? This guy right here, eukaryote. We have nucleus, rough ER, or rough endoplasmic reticulum, mitochondria, Golgi apparatus, and then some other structures here that you don't need to know. But all these organelles inside the cell working together is going to make this a eukaryote. Plasma membrane, and this is important for maintaining the shape of the cell. So bacteria or prokaryote and eukaryote here both have the plasma membrane, keeping the shape of the cell. It's also keeping things on the outside. So if there was like chemicals on the outside of the cell, these chemicals would try to get into the cell, but when they hit this plasma membrane, they would be pushed back in that direction. So it's a way to keep things on the outside from going to the inside, also things on the inside from making its way out. Cytoplasm. So if you take the, if you take the plasma membrane and you go from one side of the plasma membrane to the other side of the plasma membrane, everything that's inside of there, that is all cytoplasm. It is the solid structures plus the liquid structures. An organelle. So we'll start with L. The L means small or minuscule, and then organ is a working structure. So if you look at all these little bitty objects inside the cell, these are all going to be termed organelles because each one has its own little function similar to what your organs are doing for your body. Most of these are already covered. So again, nucleus holds the DNA, rough ER makes proteins, mitochondria makes ATP, centrioles move things around inside the cell, lysosomes digest, peroxisomes detoxify, microtubules hold the shape of the cell. So all those technically are organelles. Let's see. The nucleolus. So I talked about the nucleus. Again, this large area where you find DNA. Now the nucleolus, this is going to be the area of that little purple dot. So you got the big circle, and then you have the small darker circle in there. So that little circle is the nucleolus. And that is where DNA is activating and genes are working. So right there is where you're going to be making something called RNA. We'll get to RNA in a few days, but RNA is the precursor to a protein. So whenever you want to build a protein, you have to make RNA first, and then RNA is like the blueprint slash, we'll say, delivery trucks that are going to bring everything together and build up your protein. We talked about this last week, but a protein is the product of genes. So every cell's job is to make proteins. This is going to like determine what the cell is going to be doing. It's also going to determine what the shape of the cell is going to be. In DNA, collection of genes. So if you were looking into that, there's a specialized code, and that code is going to be different for everybody, different for every person and for every organism. And that code is going to determine what genes are going to be making for what protein. And then RNA, it's the precursor to protein. So it's called the dogma. We'll get to this later, but dogma, you start with DNA, you turn it over or convert it into RNA, and then when you get RNA, you convert that over into a protein. So these are some examples that I pulled from other sources that I use. So I'm going to show you the difference between, again, a eukaryotic cell and a prokaryotic cell underneath the microscope. So these guys over here, these are actually fish egg cells. So fish eggs were taken very early and they were dissected and stained, and this is what the cells would look like. So again, this line, a lot of people think that's the nucleus, but this line I'm drawing, that's actually the plasma membrane of the cell. So that's how big the cell is. Now, this circle in the middle, that's the nucleus. And then that's the structures you see off inside of the nucleus. Those are all chromosomes. And each one of those chromosomes has different segments of DNA. And then whatever the genes are on that chromosome is going to make a specific type of protein. And there's another one right here. I trace that one. So again, that's one cell, And then this circle in the middle, that's going to be my nucleus. So this guy right here, or both of these guys right here, these are both eukaryotes. Now, also down here, let's trace this guy. So there's roughly one cell. There's a cell. There's a cell. So all these are different cells. And then you can see these dark little circles in the middle. So each one of these dark circles, each one of those is a nucleus. Again, each one of those nucleus is going to have DNA. And these cells are actually from the inside of someone's cheek. So these are called cheek epithelial cells. So if you were to scratch the inside of your cheek and then rub it on a slide, these are what your cells would look like. They're very flat. They're called squamosal cells. But yeah, both of these, since they have a nucleus, eukaryotic. And then with these guys over here, both of these are example of prokaryotes. So these are extremely small. So this two, and then that right there means micro. So these are 2 micrometers, which is extremely small. If they were in comparison with these guys over here, they would roughly be about that size. So extremely small little guys here. And you don't see those dark pits inside of these. So these guys here actually don't have any nuclei. So all these guys are going to be bacteria cells. And then they're also prokaryotic. Same thing over here, these elongated cells. So this guy right here is actually dividing. So right there, he's divided into a new cell. And then you don't see those dark pits. So none of these guys have nuclei. So all these guys on this slide are also prokaryotic. So I talked about this one ago, but this guy right here is a rod shape. And then these right here we call cossi or spherical. So bacteria come just like our cells, they come in many different shapes and sizes. Does anybody have any questions? Be good? So, relating this back to genetics, the reason why this is important is to understand genes, you need to understand where this structure, DNA, is, and then also, too, let me clean my slide off. Oops. So you need to understand where the DNA is located and what it's doing inside of the cell. So let's focus on this guy. Last week we talked about Mendel and his genetics, right? So inside of this nucleus, what we have discovered since Mendel's experiments is we have 2 sets of chromosomes. So we'll have one long linear chromosome, and then we'll also have another one the same shape and length. And then these are going to be called pairs or homologs. And then areas on these chromosomes, they're going to have the same types of genes. So when we looked, or when Mendel was doing his experiments, there was different traits for those pea plants, right? There was pea color, or flower color, pod shape, the length of the plant. So if you would look at chromosomes, there's going to be different genes on the chromosomes. So let's see, we had G and see, was it G and G or was it Y? Y and G. So the Y would be on one of these chromosomes, and the G would be on another chromosome. And let's say we did the length of the plant. So you also had another gene, maybe it could be on a different chromosome, it could be on the same chromosome, but there was a copy on one of the chromosomes, and then the other copy was on the other chromosome. So when these split and they're given off to offspring, one offspring would say get the Y and then the T, or they would have to get the small g and the T. And it's completely random how this happens because of the way the fertilization works. And then another parent would come in and they would donate another set of chromosomes. So let me go back to here for a second. Camera in my way. So you can see this parent was YY. So that means on chromosomes, if you were to draw chromosomes out, it would be YY. Actually, no, let me do it over here. So I'm going to do that one right there. I don't know why it's doing those lines. And then we'll do this guy here. And then he was GG. So chromosome set. Chromosome set. Let me try something. I don't know why my pen's messing up like that. Mm. Okay, so look up in the corner. Again, Y means yellow, and if I get a G, it means green. Uh, Remember, Y is dominant, and then G is recessive. So if you see the Y, no matter if there's a G there or not, the individual is always going to be yellow. Only way to get green is to get two copies of the GG. So with this cross, if one parent is always YY, that means every offspring created is going to have one copy of the Y. So if this was to come down here, combine with one of these, so bring that over here, this individual is going to be YG. And then if that chromosome comes down here and this chromosome comes over here, that individual is also going to be YG. So if you were to look chromosomes, now you have one that's Y on the chromosome in the DNA. And then over here, this individual would have a G chromosome. So that would be the what's inside the cell. And again, since this individual got a Y, that means that individual is going to be yellow. Same thing over here. This individual got a Y. That means that individual is yellow. And then if you go up here to this is why Mendel found that all his individuals in the first cross were all the same color. Let's see. Where's it at? Yeah, here it is. Uh, the green disappeared. All the individuals that were created from this first cross technically were all yellow because they all had this Y gene. So if you take YY or YG as a parent and then another YG as a parent, chromosome. There's there's chances that the Y's will be added together. So maybe YY. So that's if that one and that one add together. There's a chance for YG. So that's if the Y and the G's add together, you actually get that times two. There's twice the likelihood of this one because there's that Y and that G, and then also that Y and that G. And you also get a G or G combination. And then going back to the phenotype, this individual got a Y, so it's yellow. This individual got a Y, so it's yellow. And these individuals down here, they're both G's, so these individuals would be green. And if you go back to what Mendel found, he found that most of the individuals that he found came out as yellow, but some, around 2,000 of his offspring, came out as green. So that again, that verifies what happened. The individuals had one copy of a dominant and one copy of a recessive. Everybody doing okay?
Yes,
I can do this much better on a whiteboard though than I can on the computer. Um, let's see. Two. So let me show you a video that I usually use with my cells or my cell class, just to get you an idea of what's happening on the microscopic level. Actually, I gotta stop sharing my stop share screen share. I want to do YouTube share. Yeah, you see YouTube now? Okay. So these guys are paramecium, and these paramecium they're actually being fed yeast. So both the yeast and the paramecium are different types of cells. So what I want to mainly focus on is that this is a eukaryote, and then you're going to see a large nucleus. So, see, you've been hidden there. Okay, wait a minute, it's moving. So on the other end, so right down here, it shows it again. There's a large nucleus. So right back there, and that nucleus is where all the DNA is being held. And since this guy has a nucleus, this is going to be a eukaryote. The yeast cells in the background, the little white particles, they're also eukaryotes. So they're much smaller. So you can see that they're both eukaryotes, but the size difference ranges dramatically. So uh, let's see. I can pause it, I guess. So this end on here has the nucleus. What you're seeing with the red, when the yeast particles go in, so the paramecium is actually eating the yeast. And then as it digests the yeast, we have a special type of stain that changes color. So it's going to be red here when they go in. And then when it's starting to go darker right here, it actually is being added to acid. And the acid is breaking it down, and the paramecium can digest the yeast particles. But I really want you to focus on this end. So this guy, this right there, that is the nucleus. And then everything that the paramecium needs, like protein wise, is held right there inside of that little packet. Shut up. Very good example. That large circle right there, there's the nucleus. So if you were to see one of these inside of a cell, guaranteed or determined fact that that is going to be a eukaryote. And that ignore that right there is the nucleus for this one. That one. Think of something else. So I'll show you this video, and then I'll come back to this probably next week too, because we'll look more into um, RNA next week. Okay, so what you're looking at, this is the plasma membrane of the cell. We're about to go much deeper into the cell. You're going to see all the organelles inside of the cell, including the nucleus. So again, this view is showing you all the different types of organelles. We have mitochondria, Golgi apparatus. These are lysosomes and peroxisomes. The area around the nucleus here, these are the reticulums. But that big purple sphere in the middle, that's the nucleus. And we're going to zoom in on that. And then when we get inside of it, we're going to see the DNA. So we're going inside the nucleus. Okay, so this structure is a chromosome. Chromosomes are actually only present during one specific phase of a cell. It's called mitosis. But with a chromosome, all the DNA that should kind of be uncondensed or it should be long and stringy, it's actually packed very tightly into this solid structure. So we're going to zoom in on this, and it's going to show us our DNA. So it's unraveling. And those little discs are how we wrap DNA. And then, again, there's our spiral ladder case or our double-stranded helix. And then from there to all the way off the screen over here, way back in the back, all that is the gene. And then that one gene is going to code for a specific protein. It is all about what pattern, or see the colored pattern? It's all about what colored pattern is there. It's going to be able to make a specific type of protein. Okay, so this guy's called RNA polymerase. So you can see it's unwrapping DNA. So it's kind of making it or splitting it apart. And as it does, it's going to read one side. So we don't read both sides at the same time. We only read one side at a time. In this case, we're reading the bottom side. And then as it runs across the DNA, it's going to read the code. And then the DNA is going to be converted over into RNA. Okay, so you can see these letters. These letters are the genetic code. Everybody's DNA is a little bit different from everybody else. So the bottom strand, the lighter blue, that's DNA that's being read. And then up here with the darker blue, this is called RNA. And RNA is going to be the building molecule for proteins. So it runs the entire length of the DNA until it gets to the very end of the gene. So it doesn't want to read the entire chromosome. It's just going to read what section or the little section of the gene. So this is specifically messenger RNA. I'll get to this next week. So we're going to manipulate it a little bit. You can see it disappeared. Again, that's called splicing. We get rid of the junk. We don't want to use it all. So right now, this messenger RNA is coming out of the nucleus. So DNA is kept in the nucleus, but with RNA, RNA is able to leave the nucleus. And then when it's outside, it's going to find the ribosome. It could be anywhere. It could be free floating. It could be on the rough ER. This is another type of RNA called transfer RNA. This is like a dump truck. So again, this is a little bit kind of jumping ahead, but next week we'll talk about proteins. This guy right here is called an amino acid. Proteins are just long chains of amino acids. And then with the specific types of transfer RNAs, they have a code that matches the code in RNA. And then that code is going to bring in that amino acid. So as you see, the RNA that we've created is going to be read through. So think of it like a printer. It's going to pull the paper through, or as it pulls the paper through, it reads the paper, and it prints off a protein. Okay. So again, our RNA is the bottom strand. It's being pulled through, and then there's codes here, as you can see. These codes are going to match one of these transfer RNAs with the dump truck. And then on top, with the little amino acids, they're going to start joining together. So the entire RNA is read. All the amino acids are used for the protein. And then when it's done, everything uh, unbinds. It lets go of everything. And then this long linear strand, it folds over, and then there's the working protein. So your cells are doing this right now. You do this thousands of times a day to make specific types of proteins. Run it through one more time without stopping. Oh wait, it jumped to new video. You go back to the other one. We'll do this one too, but not today. Let's see. And if it wasn't for Mendel's experiments, we would have never found this out because we knew that there was a particle to look for because of Mendel's experiments. That strand of RNA, or yeah, that messenger RNA that's being used right there, it can be reused. So as one ribosome pulls it through, another ribosome can bind a hold of the end that's free, and it could be read again. So you can actually mass produce thousands of proteins with just one RNA strand. Kind of saves you time in the long run. Then when it gets to the protein, we'll pause it. So you see how it folds over on itself and then it makes this specific shape. So this specific shape means it works correctly. Mr. Harry posted a link earlier this week about gene therapy and about an individual that was sick. So those individuals that have that specific disease, it's called cystic fibrosis. So cystic fibrosis individuals, there's a protein in their lungs that's not made correctly. So when normal humans breathe, they have a very thin layer of mucus on the lungs that allows for oxygen to go over into the bloodstream. Individuals with cystic fibrosis, they make a protein that's shaped abnormal. And this abnormal protein makes a very thick mucus. So think like mud or clay inside the lungs. And that's going to prevent oxygen from going over into the bloodstream. So they get very weak and very sick very quickly. The gene therapy that Mr. Harry posted, we can actually go back. So if we look at the code, see where's it at? So see this code here? If there's something wrong with the code, then there's something going to be wrong with the protein. So that's what those individuals have. They have a mutated or messed up code. Gene therapy can go in and we can try to fix the code to make it working right. We can't do every cell, but we can do most of the cells. And individuals that have the treatment, they just have to keep getting the treatment over and over again because since they have a specific DNA code that's messed up, they're going to have that their entire life. But if we're able to go in and treat it one time, we should be able to keep going back and treating them over and over again. And that's going to go in and change the code to working order. And then that's going to make the protein work so it thins out their mucus and they can breathe normally. So we can, again, if we can figure out why genes are mutated or why genes don't work, we can go back with specific methods that we have now, gene therapy, even CRISPR, but CRISPR is still kind of very new, and try to fix these codes to make them work correctly. And it's all very circumstantial, but we might be able to fix a lot of diseases this way. Does anybody have any questions? Yeah, so next week it looks like we'll go over DNA. I'll tell you what DNA is made of. I'll show you the codes for DNA and RNA, and then I'll show you that video again that we just looked at. So I'll plan on doing 6, 7, 8, and 9 next week because it's all pretty much all the same thing, and I'll send you the pages for 6, 7, 8, and 9. Okay. Um, I'll put together another list of terms and maybe a sample of the code. That way you can go ahead and start looking at the code. That way it's not brand new to you. So anybody have anything for me? You're able to follow me okay?
So far, it's quite difficult to understand because, well, first of all, thank you for your explanation. It's sometimes easy to follow, but sometimes a little bit difficult to understand. But
I
maybe
completely
if you
understand. Yes.
could send uh, send us some note maybe regarding any determination of the code, maybe we can slowly understand while read some journal maybe.
Okay.
For myself, I can understand some words, but sometimes I'm lost.
Yeah,
Okay.
we do have very big words here. Unfortunately, there's no way to change up those words because there's no other word that kind of can swap out the bigger words. But yeah, I can make some notes about DNA and the code, and then I might can give you some practice for converting.
Yeah, practice will be good.
Okay. Yeah.
Maybe some short quiz or something so we can digest slowly.
Okay. Yeah, I can do that.
Okay, thank you.
You're welcome. Anybody else? Okay, so if everybody's good and doesn't have anything from me, I will end for today. Go ahead and stop the recording. Let's see.